Yeah.
So, good morning, everyone. My name is Shauna Kulkarni, and I'm from Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research. And welcome to a one-day national webinar on exploring innovations in novel drug delivery system under the aegis of International Internal Quality Assurance Cell, or IQAC, which is today, 30th September, and it is organized by Google Education Societies, Sir Dr. M.S. Gosavi Pharmaceutical Education and Research, Nashik. So before starting the webinar, let's hear something more about our Gokhale Education Society. The Gokhale Education Society was established on 19th of February 1918 by Principal T.A. Kulkarni, who was a great social worker on the occasion of third death anniversary of Namdar Gopal Krishna Gokhale, who was a spiritual guru of Mahatma Gandhi. The society has completed 105 years of meaningful existence and today has more than 140 units spread across three zones of Mumbai, Nashik, Thane, Palghar, serving 1.25 lakh students. The society is committed to the empowerment of students at all levels, especially through access to a higher education to achieve world-class citizenship through relevant courses and formal and informal streams. Further, the society is committed to uphold the reputation of teaching profession and establishing a culture of care and excellence by providing a wide range of professional vocational courses for poor and downtrodden, as also for Adivasis and backwards to meet the changing socio-economical needs with human values and social responsibility. Currently, Gokhale Education Society, led by Principal S.B. Pandit, President Dr. Mrs. D.P. Deshpande, who is Secretary and Director HR, playing crucial role in the overall progression of Gokhale Education Society. So about our college, which is Sir Dr. M.S. Gosavi College and College of Pharmaceutical Education and Research. It was started in Nashik in the year 2012. Presently, the college offers PG programs, M.Pharm Pharmaceutics, which has an intake of 15 seats, and Pharmaceutical Quality Assurance of 3 seats. Then we also have a B.Pharm program, which is permanently affiliated to Savitribai Phule Pune University, or SPPU Pune, with an intake of about 100 seats, and D.Pharm program affiliated to MSBTE Mumbai with an intake of 60 seats. The college has state-of-the-art infrastructure and has a lush green surrounding, which provides favorable milieu for students to grow not only as a good pharmacist but as a world-class citizen. College has an experienced and qualified staff with academic excellence. The college maintains a tradition of good academic results and organizes various activities including conferences, seminars, student faculty development programs, workshops for overall development of students and faculty to bridge the gap of the academia and the industry. So I welcome you all to this webinar. So let's start the webinar by paying honorable homage to late Sir Dr. M.S. Gosavi, who was a great visionary and an eminent personality who rendered invaluable service of 65 years in the field of education. So welcome everyone. So we have two speakers for the day, Ms. Neetu Saudagar ma'am and Mr. Ashish Parashar sir. So I'll pass the stage over to Patil sir for the further program. Now I will introduce.
So, YouTube YouTube स्क्रीन टीव्ही घेते स्ट्रीम ऍड मध्ये चालेल.
So, hello everyone. This is Professor Deepak Patil. It's my pleasure to introduce our speaker, Ms. Neetu. So, ma'am is PhD scholar in pharmaceutics subject, and ma'am has completed her masters in Dr. M.S. Goswami College of Pharmaceutical Education and Research, Nasik. Ma'am has completed her bachelor in pharmacy, R.G. Satpal College of Pharmacy, Nasik. Ma'am has also completed her advanced diploma in RA and diploma in PV and clinical research from Elite Institute, Pune. Ma'am is expertise in medical and regulatory writing, clinical and non-clinical research, and has now currently working with product like as a medical writer. And ma'am has also worked with Blue Cross Laboratories and Denmark Pharmaceutical R&D. And ma'am is having various publications in reputed journals. Over to you, ma'am.
Thank you for this introduction, sir. First of all, I'm very overwhelmed because this is my college. I have started from attending guest lectures to giving guest lectures. And I don't know what to say about this introduction. This is really overwhelming. But starting from the start, I would like to first of all thank Boraste sir, then Pingre sir and Amrutkar sir for organizing this seminar and starting this chain of considering us of being capable enough of delivering guest lectures in our college. Sir, this is a huge opportunity, and even today that I'm presenting, I feel like I'm giving a viva. You have prepared us so well that while I was even preparing my presentation, it was like, the format should be same, the font should be similar, the references should be all in one style of referencing. So all of that comes from our staff. It comes from Pinge sir, it comes from Boraste sir. So today, as I'll be presenting as a student of Sir Dr. MS Gosavi College actually, because all these soft skills that I have developed through the process that we go through as a master's program that the college offers. And yes, also, I would like to mention that my mother and my uncle, Dr. Sanjay Saudagar and Miss Sindhu Saudagar, they're attending besides their work. So, a special thank you for attending this. And yes, starting with our topic today, let me just quickly present my screen. Okay, I just hope my screen is visible. Right. So, topic for the day is just a second. Yes. So the topic I'll be covering today is respiratory, or we call it as pulmonary drug delivery systems. And because this is a niche area, I feel like most of the students, because I know most of the audience consists of BPharm, MPharm students. While we are thinking about projects, let me jump to that end, because ultimately we have to uh, come to that part where we need to formulate something, evaluate something, and present something which is of our own. So while we are doing that, what are we thinking about? We are thinking about dosage form. What dosage form should we go for? And what are the first dosage form that pop up in our mind? Tablets, capsules, syrups, suspensions, emulsions, creams. So that I feel that somewhere we need to, think bigger. So what if dosage forms that are targeted to the respiratory system? So what that these there is a possibility that the manufacturing criterias don't match the facilities that are available. So what that the drugs that are included in this kind of a delivery system is expensive, requires a certain kind of particle size. So what? So, like, at initial stage, what is important is you need to think that this is an area where this is a targeted drug delivery system. So why not go with a dosage form which takes your drug directly to a organ and solves a lot of your problems, like the bioavailability issue is there, the systemic circulation issue is there. And also, one thing specially I'm mentioning to the MPharm students who are listening to me right now, not just our college, any college students who are listening to me. uh, you know what the problem is, that we start from tablets, we think about tablets, we decide about tablets, and then we make tablets. So the beginning is your thought. So if you start thinking from a dosage form that is targeted, if you start thinking from there, you're
Excuse me, ma'am.
yes.
Sorry to interrupt you, but you're not visible.
Oh, okay, okay.
Yeah, your screen is visible. Now you're visible too. Yeah, thank you.
Is my screen and so simultaneously
Yes,
that'll
yes,
work, right?
yes, yes. Yeah,
Oh,
it's working simultaneously. You're visible too.
good. Can I continue?
Yes, yes. Please continue.
Yes. So my point was that if you start thinking from here, at least you can work with, see, how am I going to manufacture it, how am I going to, how is the entire process going to go? That is the secondary part, because there is something known as funding, there's something known as scholarships. At least start, at least start with that thought. Once that thought is there, everything else follows. So I would really like to recommend you all that if you don't focus on different drug delivery systems right from now, then what will happen is you are limiting yourself, you are limiting your flow of work. So once we are thinking about publications, once we are thinking about, you have to at the end of the day present your final project in front of a team that is going to interview you for R&D position, a R&D position. So when that happens, what comes is you're taking a risk. You're taking a risk, as in you're going with a drug delivery system that is different but effective. So that thought, I think, if you start with that thought, I would like to start this presentation with that. That is what we started our projects with. And we are very lucky that our college supported us. We got a chance to intern at Glenmark Pharmaceuticals R&D. They helped us with these kinds of projects, and I'm very very sure they're going to help you also with these kinds of projects, provided you are going to choose drugs, you're going to your synopsis is going to support these kind of drug delivery systems, right? So with that thought, let's just begin. So see, my flow of presentation, or my thought process rather, is always like, I start with questions. So only when you bombard your own self with questions will you get answers, is what I feel. So my flow of presentations have always not been so conventional, I would say. They're always like, where I start thinking from, I put that on my slides. So what we are going to deal with is, first of all, where or who, like this respiratory drug delivery system, where are we going to use this, and who is our target audience. Then second will be, why do we need a respiratory drug delivery system at all. Then third is, what is this system. Now that you know why do we need it, I think the first question that should pop up is, what is this. That curiosity develops, right? Then after that, when you know what it is, how can I make it. As a pharmaceutics person, my first question is, how can I make this. So that is like, after that, what comes is, what's new. So this why this question is important, that what is new, is because uh, I have to start thinking about a project, right? I have to have one idea, but then that idea shouldn't matlab, it should already not be there available online. So there is no point if I'm just repeating what has been done. Then what is the novelty. Why am I doing a repeated thing? I can just copy paste from some domain, but we don't want to do that, right? We're all different. We're doing a project, we're investing 3 months, 4 months, 6 months of our time. We want to do something that is different, that is not available in the public domain, right? So this question, that what's new, what's available, what's happening, updated. Go to Google Scholar, filter recent articles from 2023. What is happening? So from that, if you skip to a good go to a good review article, if you skip to the applications part, you'll see that what is the novelty, what kinds of drugs have been used, what is the kind of dose that drug has been used in, what is the kind of population that drug has been used in. So all of these questions, when you get answers in, you find a dark area. And by dark area, I mean that you find an area where you know that yes, now there's this one area that seems to me that this drug can be used in this drug delivery system. This looks very underdeveloped to me. So you can start with that idea. So that gives you a starting point, right? So first of all, let's begin that respiratory drug delivery system, where and who. For that, this is my first introduction slide. So this is one of the videos that I came across. I found it that this video is like, sums up, is a good start for the presentation. So let's just start with this. So aerosol drug delivery systems. Aerosols, meaning, I think all of you must be aware of that. Aerosols are small air particles that will carry your drug. That will carry your drug where? Of course, respiratory, meaning the lungs. The first thing that should pop up is inhalation, exhalation. So it is an inhalation therapy basically that is going to go along with your inhalation. So what are the problems? Why at all does a person need an inhalation therapy? What might be the problem? Of course, aerosol drug therapy will be needed for people who are suffering from diseases that are related to the lung. What are diseases that are related to the lung? It may be asthma, it may be COPD, it may be abnormal breathing difficulties, or with age what happens is mucus blocking happens. So that can be one thing. So now that you start thinking that where am I using this? So you can now list down your diseases. Asthma is there, COPD is there, bronchitis, cystic fibrosis. Now I know that where I have to use this. So now I know that, okay, this is a major problem. 60% of the world population suffering from asthma. So whatever drug I'm making is going to be useful for the 60% of my audience, right? What is the effectiveness? So aerosols will have a smaller particle size. Aerosols will give an effective output that we'll see in further slides. So I hope that one thing is clear, that where and how. So inhalation is your how, and where is your lungs, of course. So starting from that, basically for every drug delivery system, there's a catch. So you have to find that catch first. Once we talk about respiratory tract and inhalation, that catch here, once you go through all the literature, or once you even start thinking about this, that catch here is that the particle size. Of course, you cannot inhale a large particle. You cannot give your patient large size particles and expect them to inhale, and again already they are suffering from blockages, tumko or block kar do. So that cannot be a solution, right? So first things first, what is my problem is that I'm going to face in future of my research is that particle size. So major problem I have identified it is particle size. So what is the particle size that I have to focus on is the question that arises. And see, why is this a flow like this is because major jo problem, what we are going to face in the research, if we tame down to that problem first na, then later on when you're going to this, are ye to particle size ही match nahi kar raha, basic criteria match nahi kar raha, so later on that question never comes. So first things first, cut off what is the basic criteria for your drug delivery system, and in this case it is particle deposition. So for that, my target organ is the lung, right? So for the lung, I need to know the dimensions. I need to know that what particles, even if with the normal air that I'm inhaling, what size of particles are reaching the lung, what size of particles are reaching what part of the lung, what part, why is this question that what part of the lung they are reaching is important, is because the diseases or the conditions that we are dealing with can be in different parts of the lung, the upper lung, the lower lung, the bronchi, the alveoli. So right, that is why we need to know dimensionally initially. So there might be studies available, people might have done studies, or there is data available regarding the physiology of the body, the lung, that what kind of particles. So one such study or one such demonstration I came across, and I am keeping visible demonstrations here because this dosage form is already very new. And if you from where I think is, if you don't see it, you don't get it. So we are going to see a demonstration wherein what they have done is they have divided particles in three types on basis of their dimensions. So first dimension is 0.01 micrometer, second is 1 micrometer, and third is 10 micrometer. So very small, medium, large. So these three particles they have taken, and what they have done is that they have observed that what these size particles eventually what how when we inhale these particles, eventually what happens to the particles as they go down the trachea and as they go down towards your lung. What happens, what are the mechanisms that happens? So we'll quickly get into this. So right, uh, this is the tracheobronchial deposition, first thing we are talking about. So this is the trachea, and this pink thing is the diaphragm. So we know the contraction and the relaxation of the diaphragm is responsible for your inhalation and exhalation. So once the diaphragm contracts, size increases of the lungs, we inhale, and once it relaxes, we exhale, right? So this is the normal breathing procedure that is happening. Now, let's see when our these are the bronchi. When our less smaller size particles enter, what happens is that their rate of deposition is high because their sizes are small. So because of that, what happens? They tend to deposit initially. Most of them tend to deposit on the walls, and some of them tend to enter inside. When the medium sized particles enter, most of them tend to enter inside because the inertia is more initially, but once you exhale, most of them leave also. So that is there, the deposition is not there because lesser Brownian motion. What happens with the large size particles? Large size particles enter, but eventually they leave. Matlab, they don't settle on the tips. They would either two things will happen with them. They will either enter, and because of depending on the gravitation and because depending on the individual's inhalation also, they will settle. So see, this is a graph that they've plotted. This is a graph which is particle diameter, which is in micrometer, which we have discussed that our particles which entered were in micrometers, and versus percentage of particle deposition on the tracheobronchial region. Now, initially, this purple line that you see is a criteria that after several researches, we have, this is the initial deposition criteria that they have marked. The blue line that you see is the nasal or pharyngeal or laryngeal region deposition that is there. Now, right now, what we have seen is tracheobronchial region deposition. Now, we'll plot that on this graph. So see, these were the small size particles. These were the medium size particles, and then these were the large size particles. So according to percent deposition, approximately 25% of the red and 0% of the medium and large size particles were deposited in this region. Now, what we'll see is now that tracheobronchial region is done. What we observe that particles between 10 micrometer to particles between 0.001 micrometer to 10 micrometer deposited. That is the range that we are looking for. Now we'll move down. We'll move down to the alveolar deposition. So alveoli, we know that alveoli work with the filtration. They tend to remove add oxygen and remove carbon dioxide from the air, and they're like branches of the tree. They increase the surface area, which is responsible for the breathing activity, or rather air filtration process, we can say. So in the bronchi, what happens is that once the air comes in and a particle comes in, there are pores inside, right? And there are small openings also to the pore, like the alveoli are micellar cells. So once the small particles come in, what happens is that because of large inertia and large rate of deposition and large Brownian motion, they tend to stuck in the alveoli. So these are our 0.01 micrometer particles. They are stuck in the alveoli. Okay. Now, what happens with the medium-sized particles? When the medium-sized particles come, the rate of deposition is low, but the inertia is okay high, but the rate of deposition is low. So what happens is some of them are deposited while some of them are exhaled due to the lesser deposition rate. What happens with the larger size particles? The larger size particles will, of course, have greater gravitational pull. So, because of that, once they enter, if due to the gravitational pull, they'll be stuck. And some of them, because we know, are already eliminated from the tracheal region itself. So, they haven't even reached the alveoli. So, whatever amount is reached, now we'll plot the graph. So, for our lesser size particles, about 43% of them have been retained in the alveolar region. Okay. For the middle sized particles, about 5% of them have retained in the alveolar region. And for the large size particles, again, a very minute amount has been retained. So now this plot that we have plotted right now is for the alveolar region deposition. Now see, what you can see from this graph is that one conclusion that we are coming to here is, very carefully look at the particle diameter and the percent of particle deposition. So your particle diameter between 0.01 to 10 micron ideally shows deposition of particles irrespective of your region, right? And once you see that as the particle size is going to increase from a 10 micrometer onwards, the deposition is almost equal to zero as far as our tracheobronchial and alveolar region is concerned. So from this study, basically what I'm trying to come to a conclusion is that my particle size for my respiratory drug delivery system should at least be in this range. If not, this is the basis of that, right? So now I have one criteria tick for my drug delivery system, that what is the ideal particle size that I'm dealing with, right? So let's move on. Right. So here's my favorite part. Why do you need a respiratory drug delivery system? Why is it that I cannot take a tablet and sleep peacefully? Why are you giving me different pumps? Why are you giving me different kinds of particles? And why am I making this? Even as a pharmacist, if you're thinking why should I make such small particles? जो दिखेंगे भी नहीं. So this why, I will tell you what happens with the bigger particle also. So with the bigger particle size or other dosage forms, what the problem is that what is the guarantee that your onset of action will be rapid? First thing. So for proving that, you have to do 10 other tests, right? That is going to consume your time as well. Okay. Second thing is that this is a targeted drug delivery system. So once your particles are in that range, nobody can actually, of course, after your evaluation is done, can question that, is this is this pump going to take that drug to your lung? Of course it is going to take. It is a targeted drug delivery system. That is how it works. Then lower systemic absorption, right? Because see, initially what happens is that if the dose of your drug is effective dose of your drug is let's say 20 mg, so because of this systemic absorption, because we know once it goes, some of it will be degraded, gastric degradation will be there, intestinal degradation will be there, that unnecessary absorption will be there. So because of that, most of it will go. So while manufacturing only 25, so that if it goes, effective dose remains. And then, but in this case that is not happening. So whatever is your effective dose, you're giving that is going to be administered, and that is going to be efficient. So because of that, what happens is that initial drug ka cost jo hai is going to be because of only your drug dose. Okay. Then lesser systemic side effects. It's as simple as that. I mean, it's self-explanatory, because if it is going to your target organ, it is minding its own business. It is not going anywhere else. So less side effects, of course. Less. Then lack of first pass metabolism. Yes, because again, same thing. All around therapy of it is conclusion of it is that because it is going to your target organ, it is going to mind its own business. It is not going to fidget with any other parts of your body. So it's safe. That means it's only dealing with the part or with the organ where the problem is. Okay. So my where is clear, my why is clear. Where? The lung. Why? Because it is targeted, it is easier, and no other dosage form can, target my lung better than this kind of a system. Okay. Next is what? So, what are these respiratory drug delivery systems that we are talking about? So, see, two things. Once we are talking about a drug delivery system, there is your drug delivery system and there is your medicament, right? Matlab, tablet is your drug delivery system, your drug, initial drug, let's say whatever, ketoconazole is there, it is your drug system. So, and when you combine both of them, that will give you a effect. So, in case of respiratory, what is there? There is a device, and then there is your drug solution. It can be a drug solution, it can be a drug suspension, it can be a drug powder form, it can be, we'll discuss that part of it later. But what I'm trying to say is that in respiratory drug delivery system, it is a combination therapy. The drug delivery will be with the help of a device that is attached to your formulation. Okay. So there are these three different types of aerosol delivery devices that I'm going to talk about today. First is metered dose inhalers, the normal asthma pumps. I don't know if you must have seen around. Then B is nebulizers. I don't know how many of you have seen nebulizers being used. Mostly they're being used for pediatric, geriatric patients. And next is dry powder inhalers. I really doubt anybody knows in detail about this system, but that is my initial idea behind this session also, that we have to, being pharmacists at least, we have to be aware of these systems, and that there is this possibility of something pairing some device with my drug so that I can take it to my lung. And you know what you can say, once you use a device for taking your drug to a specific location, it adds to your therapeutic efficacy. Ultimately it adds to the therapeutic efficacy of your drug itself, and there's lesser wastage also. So now when we are going to talk about respiratory drug delivery system, we not only have to talk about our formulation, but we have to talk about these devices also. How do these devices work? So there are two things that I'll be discussing: how these devices are working, plus what is the kind of formulation we have to deal with. Okay. So first is metered dose inhalers or asthma pumps, as in local language. But I prefer and I hope that after this presentation, all of you are going to call them metered dose inhalers. That is the correct name for these. So, as the name suggests, meter dose inhalers are inhalers which will administered a metered dose. What is a metered dose? That means a specific amount of dose in one pump. Okay. And because again, like I said, it was going to be very difficult that we only talk about it and not see it visually, that how it works. It is going to be very difficult for you to remember, but just know that this asthma pump cut is metered dose inhaler. Okay. So we are going to see a short video about how this works. Hi. Okay. So this is a metered dose inhaler. So once you pump it, the patient pumps it. What happens is that the dose goes inside the tracheobronchial region, going inside to the bronchi, to bronchioles, to the alveoli, and that is how it works. So this is the metered dose inhaler. This is a canister fitted into an actuator. It comes with a follow instruction, because see, for you and me to understand it, now I'll explain the device to you. For you it is maybe easier to understand, for me maybe it'll be easier to understand, but of course to the patient, asthma patient especially, or a aged geriatric patient, they, for them it has to be through a instructions manual. And also I would like to say that mostly corticosteroids are being administered via such delivery systems. So it's always important that even if somebody nearby your vicinity or nearby you is using such inhalers, also you should be reading these instructions so that you can guide them. That is what we do, that how are we supposed to use it, how many pumps they are supposed to take, what is the correct way of using it, what time of the day it should be taken. So yes, all of them come with a different instruction manual. Right, the better part. So it has a mouthpiece cover. You open it, then there is a mouthpiece. Then there is a medicine container that is inside, fitted, inverted. So once you, the correct way of using it is remove the cap, shake it, because if it is a suspension, you need to shake it, right? Then pump it. The medication comes out. And then after that also, after the use also, clean it, close it, and keep it. This is the full demonstration of how it should be used properly. See, the full mouthpiece section goes inside your mouth. With the pump, you're inhaling, you're taking a deep breath in. So with that deep breath in, it forces the particle, it gives it a shear so that they move inside. And then they ultimately reach the lung. After that, 10 seconds countdown should be done. And then you exhale. And then the patient exhales, I mean. So this is the correct way of using a metered dose inhaler. So now you know. Okay. Now that we have seen what an inhaler is, at least what it looks like, what are the parts. So next time if you come across an inhaler, just take somebody's container, of course, used container, unused container, don't fidget with unused dosage forms. Used container, you can actually remove the mouthpiece, you can remove the cap, you can see the mouthpiece, you can remove the canister, you can see how the can is made, what is the dimension, what is written on the can, how the can is crimped, everything you can see next time. Moving on, now that you have seen that what is a meter dose inhaler, why we know, where we know, coming down to the components of a meter dose inhaler. So like we have seen, there is this uh, this outer portion that is there, that is your actuator. This container that was inverted inside, that is your canister. Then this is your mouthpiece area. This is where the administration is going to be happening. Now what is inside it? Of course, we are not just interested in how it looks, but now what is inside it, how have we exactly filled our canister. So the canister consists of a gas phase, it consists of a liquid phase. Now this liquid phase will consist of your formulation. It may be your solution form, or it may be your suspension form, depending on what, depending on your drugs characteristics, depending on your drug solubility, depending on your drug combinations, several criteria. Then there is a metered valve here. So metered dose inhaler. How is it that only you're saying that at a time a metered dose is coming out? So that trick is here inside. This is a metered valve, which what happens is once it is pressurized, only that amount of for that amount of time, that amount of liquid or that amount of aerosol comes out, and after that it is closed. So one pump, only that much comes out, then it's closed. Again one pump, that much comes out, it's closed. That is the working, that is the metered working, we can say, right? Then this is the expansion chamber, of course, because it is streamlined. This is going to, once you press this, it is going to come in one streamline, and then there is going to be an expansion chamber which is going to give it a space to like deodorants. You can take an example of good a deodorant, like you spray it, so there's an angle that it comes out in, so it is trapped inside your can. So once you push it, it's going to come out in an angle. So that is what is exactly that mechanism happens here. So, and because of that pressure, of course, high velocity particles come in. Why do we need high velocity particles? Because they have to travel a lot, right? Now two main components as a formulator, enough of the packing part. The main content, let's move on to the main content. What components do I need to manufacture a metered dose inhaler? So there are two main components. It's a very simple dosage form. There are only two components that you need. You need your gaseous phase, which is your propellant, which is going to add the shear or the pressure that is required inside the container. And you need your liquid phase. The liquid phase is going to be, of course, your drug in solution format or suspension format. So these are the two components that we are needing. So your drug is going to be of what particle size that we have seen, less than 5 micrometer preferably. Then that is the size range that we are working with today. Then the gaseous phase, the propellants we use as gaseous phase here, and mostly hydrofluoroalkanes are being used depending on their compatibility with your drug solution or drug suspension. That is again another big topic that we'll cover some other day. Uh, Moving on to how do you manufacture this? What is the steps of manufacturing, right? Before that, this, yes. So this is the canister that you saw, which was inverted here. So if you, so it's ulta. So if you straighten, I might have someone. Okay. So if you invert it, it is going to look like this. So this is how it is. This is your formulation wherein your drug, this in this case it is a suspension, is filled, and again from that opening we'll add propellant to it. So first things first, what we do is we start with our liquid phase. We start making our liquid phase. For that, we are going to weigh our drug. So how much of a batch we are making, that is very important. Suppose my dose is 5 mg. So I'm going to take 5 mg of my drug substrate. I have to dissolve it because I need it in a liquid format. So if it is soluble, whatever solution it is soluble in, if it is soluble in ethanol, I'm going to take ethanol, I'm going to solubilize it, I'm going to make my liquid phase. Done. My liquid phase is made. Then next I have to then add what? Propellant to it. So now there are two ways. uh, Also a special mention to Glenmark R&D here, because all of this we have seen and we have observed, and these high-tech machineries that they have, that is what happens there. Formulation aspect, there are two things I'm saying. That first thing is that there is a setup wherein you can put your liquid phase solution, you can adjust your propellant, like the propellant cylinder is there, which is being attached to the entire system. This is your liquid phase, this is your propellant phase, that cylinder is attached, the mixing happens, and then through a tube, through a machine, all these cans are being filled. All these cans are being filled, and then that is how your canisters are ready. This is one. Second, like, because we are talking about small scale projects or college level projects, what we can do is we can separately make our liquid phase. We can separately fill that phase in our canister, and then we can crimp it. Like, crimping is that silver part, whatever you can see on these bottles. These are the walls that need to be crimped here. Crimped as in sealed. Easy. Sealed. So there is a crimping machine that you use to crimp it so that the propellant phase doesn't come out. And then you fill it with the propellant manually, and then your canisters are ready to go. Then you fit it in that device. Which device? The actuator, mouthpiece, entire design. You just fit it. You have that device, you have this canister, you invert it, you fit it, and then you use it. This is very simple way I've explained this formulation aspect. Now, going into the basics, like what is the kind of drug substrate that I need. See, because this is targeted, of course you have to first of all decide what disease you're targeting, what space you're targeting. Depending on that, you select your drug. Then you see to it that the drug is available in required particle size. Then you see to it that you have excipients which are going to dissolve your drug. You see to it that if it is, even if it is a suspension, it doesn't sediment very quickly, because sometimes people may not shake it. Matlab, you don't want that after one pump only it should sediment, right? Some window has to be there. Even if it is a suspension, there has to be a sedimentation window that we have to offer, right? So all of these are formulation aspects that you need to take care of once you're thinking of a formulation like this. So even if the components are less, I'm just saying that there is a liquid phase and there is propellant, and then add it and then make it. Even if the components are lesser, but the technicality is there. So that is what makes this unique, but then that is what makes it effective also. Okay, next. Before that, quickly see this. Huh. So sometimes what happens is that for pediatrics children, first times who shift from a nebulizer to a inhaler, what happens is that they have a problem with the entire process of the small mouthpiece is there and then the inhaler is there, so that gap is very less, so they are having an issue. So for solving that issue, some metered dose inhaler come with a spacer. So this spacer is like a tunnel, you can see here. Let me play this. Okay, so what happens is that once you attach the spacer to your mouthpiece and pump it, the spacer gives them a time period, you can say, like from मतलब झटके से नहीं आएगा. You don't have to pump it, inhale it in microseconds. So that gives them a window. So because of that, the patient compliance increases. Okay. Then again it comes with a manual, and then again you have to, of course, if now you are using a spacer, then there are chances that there'll be deposition. So in that case, what you can do is you can use you need to wash the entire spacer after one use. So once you use it, you wash it and then you reuse it. I'm so sorry, just give me a second. अहो माझं प्रेझेंटेशन चालू आहे. हां, ओके. Okay, I'm so sorry for that. I'm so sorry. So where were we? Yes, we need to wash the spacer and reuse it. This is our canister. This goes into our actuator. This is a spacer. Attach the mouthpiece of the canister to your spacer. Open it. Okay. Skipping this. So this is one way of, easing the patient compliance. That could be one problem. This we have gone through. Moving on to evaluations. See, any formulation, of course, you need to justify it. And if you're formulating it in no matter what environment you're formulating it, you need to evaluate them. So these are certain, not certain, all, in fact, according to the FDA, whatever parameters you have to evaluate for an metered dose inhaler, all these I have enlisted here. So basics, I'm cutting the basics which you might just understand by reading. Appearance is there, microbial limits are there, water or moisture content is there, dehydrated alcohol content is there, net content, drug content, impurities, all these are analytical testings that will be happening. Particle size distribution is one important criteria, like I said. Spray pattern, plume geometry, these are new words. Leak rate will be tested, pressure will be tested, wall delivery will be tested, and leachables will be tested. But I want to focus on these two evaluations that were new to us also. I'll just support that with a figure. See, so once you're saying spray pattern or you're saying plume geometry, first of all, what is a plume? So once you spray your inhaler, you pump your inhaler, your suppose I'm pumping it from here towards you. So if I pump it, the formulation is going to flow in this way. So the formulation is going to have a certain angle, right? It is going to have a certain plume height, what we call it. So this is the plume height. That what is the range of your what is the range of your formulation reach? Then this is your height, this is your plume width. Now why is this important? That is at all your formulation reaching, the required, is giving the required pressure that it goes inside. So that test is very important. These are one of the most required tests, if you can say, like, if even if you're formulating this, you for publication you need these testing. What is the spray pattern? So once you spray anything on paper, also suppose spray your normal deodorant on paper, whatever area is being covered, that is your spray pattern, that is the spray's pattern. Of course, from a specific height, guidelines are there. From a specific height you spray, the height increases, and you observe ki kitne distance se what is the spray pattern that is happening. So for by that, what we come to know is how much of area is being covered. Okay, then so this is the spray pattern ultimate result from lasers. If you observe, this kind of a pattern comes in, wherein what we do is we take the diameter, the maximum diameter and the minimum diameter, and then we tally, like, for 10 short sprays, 20 sprays, what is the changes in the spray pattern, and then we come to a conclusion that yes, this is compatible. Even after 10 sprays, the spray pattern is uniform. Okay, next is these are some marketed formulations that I have enlisted. It's a very common marketed formulation. Fluticasone is there, budesonide is there, mometazone is there. So these are there, available in market. Moving on. Nebulizers. So first of all, what is a nebulizer? It is not a steamer. It is not a steam taking machine. A nebulizer is another inhalation therapy device that is going to have, that has different components. So see, we have discussed one device. So the metered pulmonary metered dose inhaler was a device. It was an actuator, it had a mouthpiece, it had a cylinder that was inverted, that was fitted. This is a setup, this is a device setup. Now we are moving on to a different device setup, which is a nebulizer. Now the question comes at that, if I have an inhaler, why do I need a nebulizer? मतलब टारगेट तो लंग को ही कर रहे हैं ना, why do I need a nebulizer? But then you're only thinking about adult patients if you're thinking about an inhaler, because pediatric patients, 1-year-old or 2-year-old, how are you going to administer? How are you going to tell that kid that you have to pump and then inhale and then hold? He's not going to follow any of these instructions. So that is not possible. And also for geriatric patients, it's not possible. Also for patients who have a disability in understanding, for them it is not possible to have this flow of guidelines to follow for that a meter dose inhaler requires. So we can say that a meter dose is very ideal for adult patients. But now when we start thinking for patients in the ICU, or geriatric patients, pediatric patients, disabled patients, even patients with Alzheimer who will not be able to have that motion activity flexible. So for such kind of patients, we need to think of a different dosage form that is going to target your lung, that is going to take your dosage form to the lung, definitely. But a device adjustment has to be made. That is why nebulizers. Now let's get into this, that what are the components of a nebulizer are and how we need to use it. Quickly play this.
Compressor itself.
Okay. So this is a compressor. That there's a tube that comes with it that you need to attach to the compressor, which is going to generate the pressure. Once you switch it on, then the tube is going to be added to a drug holder, or drug component holder cap. See, there's an attachment. You have to attach that tube to that drug component holder. Is making sure that all the fittings are okay. Otherwise, there are chances that it might leak, or the pressure will not be even. Then there are 2 mouthpieces that come with it. This mouthpiece is basically for pediatrics, because you can easily make them wear the mask, and then the procedure keeps on happening. And the other one can be used for adult patients also. So if you have a nebulizer, you can use it like for both the patients. And there is this component holder also, so you can carry it, all components inside the compressor itself. So what happens is that this drug holder component that is there, that you can attach that to your mouthpiece, this one or the other one, like I said, the mask for pediatric or ICU patients which we use. So this is the entire setup of a nebulizer, right? So now what, see, so our area of interest of course lies in this drug component holder. So how am I going to put my medicament inside this, because it is hollow, and every time how am I going to put my component inside this, or what is the nature of my drug or my formulation that I need to put inside this, that is an important question, and that we are going to see. So there's an opening, it's like there's like a cap and there's like a base. So in the base you have to put your liquid from the sides, using a syringe you have to put your liquid formulation that consists of your drug. From the sides you have to pour it inside this component. So, and you have to pour it and hold it upright, because of course if you don't hold it upright and while you're even administering, and if you tilt it thoda sa bhi, it is going to fall because it is a liquid formulation. Then you're going to tighten the cap, the upper cap that is there. Of course he's for showing he's tilting it, but ideally you have to hold it upright and then do the entire procedure as it is. Then you have to attach the tube that is already attached to our compressor. That tube you will be attaching here. Then yes, right, now this part is ready. Now what happens is that, because the once you switch on the compressor, the pressure will be applied, and then because of that the airflow will be there, and then your whatever your medicaments or whatever your liquid solution will be there, that will be, because of that pressure it will be pushed inside, and from the mouthpiece you can inhale it, or you can attach in case of pediatrics, like I said, you can attach this mask. And yes, you have to be very sure that you don't tilt it, because otherwise the drug formulation comes out. If you have, sometimes the screw, if it is not tight, there's a chance that the formulation comes out. He's very yes. So this is the ideal setup for a pediatric patient. A nebulizer setup for a pediatric patient that will be attached. So now we'll see that how are we going to add the, so here he has taken a saline solution for demonstration. So a saline solution he's going to add to our nebulizer drug component container. See, you have to hold it upright, you have to fill your syringe with the drug, you have to pour it. He's already poured it in, so he's just showing that how much of it is poured and how much of it you have to pour. Of course that depends on your drug. One time inhalation mein kitna drug chahiye, that much you have to fill in there. So that, because yes, mostly these systems are used in hospital setup, or in pediatrics the mothers are trained. So then you have to pack it, hold it upright, and itself you have to pack it, and then start. So see, that is how the steam comes in, and that is how, so you see the pressure that with which the steam is coming out. So once you attach it, of course, to your mouth, and the medicament comes out with that pressure, of course it is going to go inside your lungs, and the particles are going to deposit, and ultimately it is going to be a effective therapy, right? So this is the correct way. So you're not supposed to talk while this, but then he's just demonstrating, so that is why he's opening his mouth. Otherwise only deep breaths are suggested while the entire procedure is going on. So this is how a nebulizer setup is. Basically this is how a nebulizer works. So right, so what he's trying to say is you cannot tilt the entire system while it is going on. A person has to sit steady or with support. In case of kids, they are supposed to sit with support, so that this formulation, this entire procedure happens properly. So this, after being used, okay, he's also demonstrating with the other mouthpiece which is being used for adults. So that is the mouthpiece for adults. So that also is possible. Like both can use it, a different provided different mouthpieces are being used. Again, same mechanism. While you're doing it, you inhale, then you hold 10 seconds, and then you exhale. So this is the entire mechanism with this device setup also. And now what happens is that once all of this is done, both all these components need to be cleaned, and all these you have to make sure that you have to dry it also before you use it second time, because there's a lot of chance of dose deposition or drug deposition is there, right? So yes, okay, bye. Yeah, coming to the formulation. Now that you know how a nebulizer is, this diagram will be very easy to understand. So this is the your drug component ka chamber that we just saw, that white part which was used to open where our drug component was. So this is that. Okay, so this has our feeding tube. This is the feeding tube from where we attached the feeding tube, right? So this is that inlet, and this was the mouthpiece outlet. So this is the outlet. Basically, this is the piece where we poured in our medicine. This is the liquid reservoir containing your drug. Then what happens is the mechanism of the device working is being shown here. You attach your feeding tube here, and what happens is there is a small cavity, because of which once your air compressor throws your air in pressurized format, because the diameter of the tube is lesser, there is a small opening. So what happens when something comes out, something in pressure comes out from a small opening, it is going to burst out, right? That is what happens. So all these compressed particles of air which were now moving in small tube areas in pressurized condition, now they're going to burst out. So what they're going to burst out, and what is going to happen is that they're going to create a shear. So because of that shear, what is going to happen is they're going to take our liquid reservoir system with because this is continuously, air is coming continuously, continuous air coming, putting pressure on your liquid and taking it up. This entire system goes on. So because of that, your liquid is going to come out of your mouthpiece. And additionally, when we are taking, we're also inhaling air, so negative pressure, so that pressure is generated via like inhalation also, and the compressor pressure also together. So the outlet is having a lesser diameter, the inlet is having a lesser diameter, inside the surface area is more. So basic physics happens, and the medicament comes out in pressure. uh, yes, I have added this one note here, that the interplay between the dosage forms and the specific type of device for aerosol generation determines the effectiveness of drug delivery in nebulization therapy, thus requires extensive understanding and characterization. Meaning that you have to understand that this liquid reservoir containing drug system that you're working on is compatible enough. Compatibility meaning, see, it it has to be thermolabile substances that you're using. Otherwise, with this pressure that is being generated, there are chances that the air is going to heat up. So it should not degrade your drug, right? If it is degraded, what is the point of taking the whole thing inside your lung also? So that degradation should not happen. So what you can do with formulation, here this is where your pharmaceutics person's roles comes in. This is where your novelty comes in. This is where you can work on. This is our area, I can say. So I like these kind of formulation systems which question you what you can do now. This is the challenge. So these kind of systems are going to make you think what I can do. There's this problem, I have to uh, I cannot change my drug every time. That is not the solution. Here, let's just change the drug. This is not thermolabile. Let's just change the drug. That is not a solution. What you can change in your formulation, that is the beauty of a formulator. Well, how can you add components, because it's a reservoir, you have to make up volume. So in order to make up my volume, if I can add something inside my drug that is going to protect it from all of these things, then magic. matlab your drug also goes and your formulation also stays, your volume is also made up. So that is the kind of thinking that such kind of formulations require, and that is what I'm trying to promote today also, that there is so much that we can do about such thing. There are so many drugs that are avoided only because they are not being fitted inside some criteria. No drug ideally fits inside any criteria. You have to make it fit inside a criteria. And especially for any all the MPharm pharmaceutics students that are listening, it's your responsibility now to take up such challenges or think about such things. Ki, nahi, I have to use this drug. I'll think about it. How I'll use this drug in a different way. How I'll do justice to this drug. If this drug is available in cheaper price, or this drug is available in my college in quantity, bulk quantity, why can I not use this drug and make something good out of it, right? So yes, nebulizers mechanism done. You have seen how it works. Next, the formulation aspect, like I was talking about. There's so many challenges that can come while formulating this kind of a drug delivery system, and mainly with liquid formulations. Once we think about liquid formulation, stability issue comes into our mind. कि अभी long term stability का क्या है and all. So for that, you need to, like, this is of course a big risk, this is of course a big jump when you're dealing with such formulations. But then you have to the easiest way I can suggest is that jot down what problem can come. pH can matter, osmolarity can matter, surface tension can matter, viscosity can matter, thermolability can matter. Once you have problems ka list na, then you can see, okay, चलो फिर pH के लिए I'll add some buffer, osmolarity के लिए I'll add osmolarity enhancing decreasing agents, swelling agents, whatever. For surface tension, let me see what polymers we have. So that kind of a thinking, that problem solution based thinking has to be there, rather than always looking for a key, this drug is only not supporting me. So the drug is not going to support you. Your formulation, other components of the formulation has to support your drug. The hero ingredient you cannot change always. You have to change the other ingredients. That is the work of a formulation person. That is the work of a pharmaceutics person. So I think that yes, so this one figure I found, this is a very good demonstration that, see, they're saying that what a antibody body stability antibody administering via nebulizer was that was the study about basically. So they're saying that the solution properties that are basically required, so pH, osmolarity, so there are certain guidelines that we have, there are certain limits that we have. So once you go through that, you of course once you have a drug formula, sorry, once you have a drug that you're dealing with, and once you have all these criterias, you can use your excipients such that these criterias meet your drug, right? So this is there, the solution properties they are saying that you have to think about. And then the antibody stability, the aerosol characteristics we have almost discussed a lot of them. Mostly the particle size is there, the deposition rate is there, then the which device you're using for this, that is there, what population you're targeting, that is there, then what is the disease you're targeting, that is there. So that size of particle you play, the tracheobronchial region, alveolar region, that space, there's some percent variation of lesser size, medium size, and larger size particle, that adjustment is there is a space of adjustment we can make, so that window remains. So this is, so I'm just throwing ideas. I hope even if it is useful for one person, I think this lecture is a success. Moving on. Okay, to our third device that I'm going to deal with today. Now, inhalers first we took, which was for asthma, which was a very known like device. काफी लोगों को, everybody knew कि अच्छा, these are the inhaler pumps that are being used. Second, we took nebulizers. That also I think maybe not most, but many of you might have known क्योंकि pediatrics में that is being used. But dry powder inhalers. So dry powder inhalers are is one of the inhalation therapy device that is going to take your, as the name suggests, dry powder inside your lung. So these are devices. So see, it's not that always your formulation has to do the trick. The device also can help. So the beauty of respiratory drug delivery system is that your device is going to help in your therapy, provided you use your devices well. First was uh, meter dose inhalers. What was there? Suspension solutions could be used. So a drug that can be made into a solution or suspension form, that that has to target the lung, so that matlab liquid formulation can be made, provided it is stable with your propellant, provided it has that drug particle size, it tallies with all, it ticks all your marks. Second was your liquid, but in second case, like you must have observed, there is no propellant pressure, so there is no combination, what we can say कि long term kuch aise negative effect that is curtailed there, so that you can use in second case, because liquid mist comes out in second case. In first case it was a pressurized aerosols are coming out. So all these formulations, see, not just that they're liquid formulation, but the devices, you see what the devices are doing to your product. So that is the beauty of this dosage form, that the devices are adding to your administration. This can be used in and why just for lungs? This can be used anywhere, provided you devise devices that can be handled by the patients. So if not that, you can, this is like a what we can say, you can work with not just your formulation aspect. Of course you have to work with your formulation aspect, but not just that, think out of it, think about how can I administer this differently, how can I make a spray out of it, can I make a inhaler out of it, can I use the dry powder inhaler container for administering my powders, instead of just filling it into the capsule and giving it to a patient, can I do this? So, and why do this is because this is a targeted delivery. Again, your systemic absorption, your first pass metabolism, everything gets cut off, right? So your efficiency of the drug increases, and ultimately we are all working for a efficient drug product. Okay. So liquid formulations we have seen. Now, what if your drug is in dry powder form? Now, why you will have this question: ki, why are you keeping your drug in dry powder form? Itna kyun? Because not all drug powders will be stable in liquid formulation. They might agglomeratize, they might aggregate. So there are different properties. You try with different drug powders, you try putting them in different solutions. Not every drug component is compatible with a liquid excipient. So for in that case, what can I do? So in that case I can work with dry powder inhalers. This is another mechanism कि मतलब अभी कुछ भी मतलब liquids are not working, then I can formulate a dry powder. But even if a dry powder inhaler we are talking about, it's not that ki you're taking your drug powder, you're putting it inside your capsule, you're putting that capsule inside this device and you're using it. Of course you need excipients, because to make up that volume, to make sure that your drug particles are exactly going inside, that specific dose of it is going inside. Okay. So that is why dry powder inhalers. What are dry powder inhalers? First of all, let's see that what they are. Then we'll discuss ki mechanism and apna formulation ka part. Okay. So see, uh, these are all different kinds of dry powder inhalers. They are devices wherein the dry powders in the form of capsules are being put inside the device. The device works in such a way that it is going to crush your capsule and it is going to blow that uh, powder content in your as you inhale. So there's this device, she's showing a demonstration how to use it, because I feel that most of you must are not aware of such kind of a device. So see, the what she's holding is a dry powder inhaler. It has a capsule inside it. That mechanism we'll discuss, that what happens exactly, how it happens. But then let's just see this demonstration, that how it works. So there is a capsule inside, and she has opened the lid of the container. Okay. Deep breath in and out, clearing the air pathway, and then taking one deep breath in with your dry powder inhaler device, holding it for 10 seconds. 10 seconds magic, all inhalers. Giving it space for deposition, because turant if you exhale, like we saw in the first video, the particles tend to come out. That's it. Administered. So again, she has closed it and kept it. So this also what they're trying to say out of this is that once you're using dry powder inhalers, after the use, what happens is there is, because it is powder, some part of it is going to deposit in your tracheal pathway, also pharynx, larynx. So that taste comes out. So for that, what you can do is, after you have taken this, after 10 seconds of a space, you can gargle with water, and then you can proceed. But then you have to give yourself that 10 seconds of space after you have taken SNA key, immediately you do it. So see, इसमें से you can get your formulation का ideas कि I need a powder that I know is going to somewhat percent is going to give a mouth feel. मतलब it is going to give a patient a taste. So you cannot keep it very bad कि वो फिर वो 10 seconds भी उसके लिए impossible हो जाए लेना. तो in that case, what you need to do, you have to add an excipient to your powder, which is going to make it better, which is going to make the person feel better. So that is one excipient you'll need here. A sweetener maybe, which is more sweet in less quantity. That you have to find out. There are sweeteners that are available. Even while we were doing our projects, we thought कि अभी ऐसे तो इसमें तो फिर 500 ग्राम तो स्वीटनर ही डालना पड़ेगा, फिर इतना 5 mg of drug and 500 ग्राम, फिर इसको तो how will we formulate this? But then no, that is what I'm saying. If you do research, there's some sweeteners even in mg, even if 1 or 2 mg you put it, you are मतलब you particles are so small. That is the beauty of it, the formulation beauty of it. As I was saying, the particles are so small that once you inhale, you'll get sweet taste pehle hi. So such kind of sweeteners are used in such kind of formulations. So that is never a problem. The taste is never a problem. Just that you should, your research should be strong. That yes, if such sweetener is available, where is it that I can get it from? Where are such kind of dry powder inhalers being manufactured? Can I write them a mail requesting that if they can give me at least 5 mg of the sweetener, so that at least one batch of mine comes out, or one sample of mine comes out, so that I can present it in front of my in front of my team. So such kind of things will happen only when you take a risk of working with such formulations. That is one thought behind it. Okay, moving forward, let's see formulation aspects. Right, my favorite part: how do I formulate dry powder inhalers? So like I said, your drug is in powder form, so it is going to need a carrier, or you have to formulate it in the mechanism of formulation you have to work on. You have to formulate it in such a way that the drug remains intact, or like the dose is being served in one inhalation, that one dose goes. So for that, these are three different what we can say procedures that are being followed depending on the characteristic of the drug, of course. First is carrier based technique, which is a large carrier is taken, uh, which is usually lactose, and the drug is being blended with it. Like you can see, a sheet is being formed once the shear is applied. What is the shear? Force or pressure. Stress upon force is applied, it disperses, and the drug comes out. So where is this shear coming from? The shear is coming from your device. The device is responsible for this shear. And what we are making is this powder formulation. This powder formulation, now there are three different methods they have given here. One, now we have discussed, one was carrier based. Second is micronized drug. Micronized drug aggregation you can use. So your drug is in micronized form. Why? Because less particle size, less than 5 micrometer we are dealing with, we need deposition. then controlled aggregation is done, aggregate is being formed. And now see, now that you are aggregating the particles, matlab फिर से the size is going to increase. So for that, initially he take micronized drug, so that even if we are controlled aggregation is done, the output size that is going to be there is going to be close to 5 micron. So that is not going to, so even if, so if initially only I take my drug which is in 0.001 micrometer, and then I try to aggregate it, so 5 micron tak there will be better aggregation, right? So but initially if you take a 5 micrometer drug and then you try and aggregate it, then it is going to cross your first basic criteria of your particle size. So that is why micronized drug is being taken. There is a controlled aggregation that is being done. Again, the shear, your device provides the shear. The mechanism of the device is such that it is going to provide shear. I think I have added a slide on that. Third is spray drying, easiest. To take a drug, spray dry it, and the voids are there wherein the drug particles are being deposited, and as the shear appears, it bursts, and then the particles can be inhaled. So three basic techniques: blending, aggregation, spray drying. Three techniques can be used for such powders. And what you have to do, what you can do is you can uh, select your drug, you can aggregate it, or you can spray dry it, and you can put it inside your device and let it disperse. Do your evaluations and let's see what happens. Next is the formulation aspects. The formulation aspect of, okay, let's play this quickly.
Drug.
See, this is the carrier aggregation technique that they have shown. These are the drug particles on your carrier particles. Once the shear is applied, the drug particles move, while the carrier particles, because they're the larger particles, what is going to happen is they're going to be stuck in the pharynx larynx region, and then the drug is going to move inside while the carrier remains here. So the carrier can be a sweetener, lactose, everything. Okay, so this was because this MDI came in between, because they are trying to say that the MDI has propellant, which is of course harmful to the environment, and to some extent, of course, in anything in large extent is harmful to us also. So compared to that, this technique of dry powder inhalers is better. So that is there. So yes, this is another study data that they have presented, where the dry powder inhaler resistance and optimal peak inspiration flow, they have proved to be, there is a net deposition that happens from dry powders also. SNE ki all of your drug is waved away from your pathway, it does reach the required regions. And yes, a medical advice is needed to use a dry powder inhaler, because first time the person needs to know that where the capsule needs to fit, how it needs to be adjusted, and how it needs to be pumped. So considering all these parameters, it is effective, efficient. All of this you have to see, all of this if you tick, then it is a good, we'll consider it as a good dry powder inhaler, right? Okay, so like we discussed in the start, that the recent advancements we need to know before we are dealing with any dosage forms. Ki what's new, what's happening, so whatever we have discussed was related to the formulation, how it is, what it is, where there are marketed formulations available, of course. What are the recent advancements? So while I'm again very thankful for this seminar, that while I was doing some research on this, I came across these formulations. I came across some smart inhalers that people are working on. I came across this ARX, dose counter inhaler. The first thing, what this image which you can see is of a dose counter inhaler. This dose counter inhaler is meaning ki there was a problem. See, there is a problem with a inhaler, that how many pumps you have made. कैसे पता चलेगा कि now it's over or it's going to be over? क्योंकि हमेशा there is propellant, तो it is going to wait. तो अभी कोई इतना, because normal patients are not going to wait on machines and see कि अभी weight कम हो गया है. You cannot expect that from patients, right? Or not everybody is that aware of. And asthma attack is, or asthma is a condition wherein which needs तुरंत, मतलब rapid administration. It cannot wait, the patient cannot wait. So the inhalers have to be promax active for that. So such kind of dose counter inhalers. So once you suppose this is an inhaler, usme there is 200 is written, that means that there is 200 pumps are available now. So once you pump once, it'll change into 199. So again, if you pump 198, so it will show you that how many pumps are remaining and when you need to refill. So that refill, what you say, warning it will give you, so that a person is aware of that. Yes, okay, today I cannot carry this pump because there are only 2 shots left. I need to carry a new one also. So that pressure it takes off from the patient. This is a very good, what we can say, like I was talking about device changes. Now this is a very good device change that is there, that has come up. Then there are smart inhalers, like the name suggests smart. While I was reading about it, but then I thought ki, if even if I'll explain it now, you'll not understand ki exactly how smart is too smart this inhaler is. So I have a video that I came across again. So see, he's using a smart inhaler. Okay. So a smart inhaler basically comes with a chip. So a smart inhaler will have a chip which is going to record the activity of not just your pump, but it is going to also record the activity of your inhalation. Whenever a patient is inhaling, what is the rate of inhalation? What is the what you can say, what how much amount of dose has been taken in one inhalation? How many? What is the, so that what happens, so after 50 inhalation, how are you going to, how is a physician going to know कि बाबा he is using a inhaler, but how much of dose is he exactly taking? कि उतने ठीक से, तो कभी कभी लोग क्या कहते हैं, घाई में they are not going to pump it पूरा, half pump हो जाता है, तो फिर what happens in such cases? So all of that, ऐसे बहुत कुछ can happen, like it is very what we can say, risky also, right? So for that, this one solution is there, that they're putting in a chip inside an inhaler and see what happens. This is a chip wherein these are 2 sensors that they have added. This is of course device advancement only, but then even if you are suppose making a dosage form and you have these questions कि ये तो normal questions आ सकते हैं ना कि what is, baba, this is so risky कि how do I know I have inhaled? Some people are like that कि continuous they're thinking कि maybe पता नहीं लिया था कि नहीं, कितने डोज लिए थे पता नहीं. So that for such kind of a situation, these kind of inhalers are being designed. That chip they have attached nearer to the mouthpiece. This is just a demonstration piece. Of course, that chip is not going to stay here, but because he's demonstrating, he's showing an open model key. There is this chip that we have attached, and then that is attached to your mouthpiece, and then that is attached to a device. Now this again, I'm saying that is because this is a demonstration, it is a bigger, it looks like a a full setup situation here, but then it is going to be only your inhaler. Whatever is going to happen inside your inhaler. So skip to the good part. Okay, so he's all set up. He's attached that cord to his computer device, and now let's see what we've got. So this is the software basically that runs. Now very quickly, wait. So see, let's just focus on the graphs. The flow temperature is being recorded here. The elapsed time, with time the flow temperature is being recorded, because of course this is like a personalized medicine. So it is different for different individuals, and this record is going to be of course, drafted and then compiled to a file and sent to your physician, so that if there is, so your health will be better recorded. Like somebody has asthma from very early on in life, so then their thing can be recorded, and then we can maybe come up with a literal personalized medicine that can be most effective to one person. Now, also, yes, I would like to mention this, that one thing is there, the cost. The cost is a very big issue here. Like now that I'm speaking, all of you also know softwares are difficult. I mean, softwares are expensive. But then, because of that, can we stop making such progress? Because this is needed now. Like we have, Chandrayaan ka, after this Chandrayaan thing, even my perspective has changed. That even in the lowest budget, they have taken us to the moon. If they can do that in pharmacy, we can also do this. What is the point of limiting that thought? Ki no, Baba, this is a costly situation. Let's just make one tablet. De dete hai, let's just finish it off. Why are we also not thinking like this? And that is why I need that thought process. Once it starts now, once it's here, it'll be on paper also. So yes, so don't have that secondary thought. Even if it's coming, why not just question yourself that why not? At least try, we'll see now. People are ready to fund such projects if you have such ideas. So they have attached this to a software. This software is going to record your system flow with elapsed time. See what has happened, what he's doing. He's showing the live demonstration of what happens while he simultaneously inhales inside the software also. So see, first inhalation there. So after 5 seconds, so from the 6th second to the 10th second, there was the systemic flow. After about 14 to 18th second, there was another flow. And after 2 seconds of gap, again. Yes. So now you see what has happened here. What the graph, if we curtail down, come back. The graph, if we come down to, what has happened is that now you can see that how much of system flow has occurred in how many seconds. So in 2 seconds of interval exactly, he has taken 3 different pumps. But the graph, you can see that initially the inhalation temperature was lesser. As the time moves ahead, the inhalation temperature rises in his case. So with this, what we can see is that the pumps have to be such that even if there are 3 short pumps in a go, so even after the 3rd short pump, the temperature is a very crucial thing here. So like that, personalized medicine, when I'm talking about, or controlled drug delivery, what I'm trying to say, not controlled as in ki dose controlled, but controlled as in ki monitored drug delivery system. So that can be imbibed into a simple inhaler by such kind of processes and can be recorded, and then we can come up with, we can also come up with different physiological procedures, that why certain things happen. Maybe just come up with another different theory also, why is this happening. This was one of the very good advancement that I came across, smart inhalers. And yes, I think yeah, I think we have reached the end of this seminar. So these are all the references that I have used. So any of you, if you want to go through anything, you can take a snapshot, or maybe I can just share this presentation with sir, and you can have this access. These were all the references that I have used, and the YouTube links are also there. In case you want to go through the videos again, you can definitely. So thank you for listening. We are open to questions, or maybe, yes, where are we? Okay. So I'm open to questions. Yes, of course. Are these products sterilized? If yes, are they sterilized terminally, or are they managed? No, they're manufactured in aseptic conditions. So before manufacturing, only the area is sterilized. So that is one thing, because like like we say, it is a targeted drug delivery system. So once we are talking about targeted drug delivery system, we cannot risk sterilization here. So initially the manufacturing process is only sterilized, and I think I have answered your question. And yes, after the pump, yes, if the question comes that after one pump, if the sterilization you're looking at, then in case of the nebulizers, like I told you, what is done is na ki the saline that comes with it, or the saline packs generally are used. So once we run 1 or 2 rounds of saline, then we are sure that it's clean. And in case of inhalers, so it is because the valve size is itna small ki the contamination is impossible. Inside piercing can be only done pressurized mein. So impurities, of course, unless and until you're using it for like 2-3 years, then that is a different condition. But we, of course, initially do this. Which are most important evaluations and which are used for drug safety purposes? Okay, I'm getting the first half of this. Good question. At least you're thinking about this dosage form. Uh, which are most important evaluations? Like I mentioned in the case of meter dose inhalers, the importance is of course ki how well is your formulation, uh, the long term stability of your formulation, the particle size, then the plume, the spray. Because see, your device is equipped, but is your formulation well equipped ki it is matching with your device? You see, the interplay has to be there. Your formulation also has to be in that stable condition that it matches your device's technique. So that all these are important. And basic, so we do the basic HPLC assay procedures that has to be done for the drugs, that is there initially. Next.
Okay, ma'am. Thanks for your wonderful guidance
Thank you, sir.
using various photos and videos you have shared regarding respiratory drug delivery systems. So I would like to share one feedback from Miss Gayatri Shinde. So webinar is highly informative. It covered key aspects effectively. The engaging presentation provided valuable insights of respiratory drug delivery system worth attending for in-depth knowledge.
Thank you, Gayatri. Thank you. Means a lot.
So, second, ma'am, thank you very much. And
Thank you, sir. Thank
accept
you.
our digital certificate as a token of love. Okay,
Thank
we
you.
are ending our first session. So we will start our next session within few minutes.
Thank you.
Thank you.